Exosome role in median epithelial mesenchymal reactions. Definitions. Exosomes are extracellular vesicles first described as such 30 years ago. Enzymes implicated in cell-cell communication and the transmissions of disease states, and explored as a means of drug discovery. Yet fundamental questions about their biology remain unanswered. Here, I explore what exosomes are, highlight the difficulties in studying them, and explain the current definitions and some of the outstanding issues in exosome biology.
In health and disease, our major goal is how do cells secrete molecules that influence other cells in a good way or a bad way. Exosomes collectively are a very powerful way and a new way of thinking how cells communicate long distance.
So when we're talking about exosomes, we're talking about really small bubbles that are released from cells.
It's much smaller than a bacterium, for example.
And you know that they are only about 100 nanometers in size, which is about 1/1,000,000th of the size of a hair.
Exosomes are blebs that are released from cells, and they end up outside of the cells and can be isolated from the liquid.
It's been found in all mammalian cells that have been looked at. One of the problems with exosomes is that they're very heterogeneous. There's very different kinds of vesicles, and it's been difficult to come with a definition that encompasses all the different types of vesicles.
If you want to kind of envision what we're dealing with here, I guess maybe you should see Manhattan as one big organism with all these different cells. Every building is a cell, and then how do these buildings, how do they communicate with each other? And that's through these small objects that are running around through these streets. When you look here from the 28th floor, you see all these taxis driving around, and but they're so small you can hardly see them. And I guess when you go really downstairs, perhaps when you take a big microscope, and all of a sudden you see that a taxi is much more complex than just a yellow dot. Actually, there's a lot of things going on in there.
Exosomes are supposed to be very powerful mediators. They carry all the signaling and the machinery necessary to go and change the behavior of a neighboring cell.
There is genetic information in these vesicles, meaning that they're not just getting to these vesicles, but when these vesicles get taken up by other cells, that they have a biological activity.
Our cells produce large numbers of different small RNAs. They're called microRNAs, and they regulate about half of all the genes expressed in our cells.
Now, we did find RNA in the exosomes probably around 7 years ago.
The major question in the field is, do cells communicate with other cells in the body by secreting exosomes carrying these microRNAs and then affect the metabolism in other cells?
Only 5 years ago, the principles of communication in the body were either hormones or immunology or neurology. And now we found another way of communicating and sending genetic signals between cells.
How is it appear in the medical part of dentistry? The formation of dentate pulp involves complex epithelial mesenchymal interactions between herrings, epithelial root sheath cells, and dental papilla cells. Earlier studies have identified some of the regulatory molecules participating in the crosstalk between hairs and DPCLs and the formation of dentin pulp. Pros and cons. We also know that exosomes are found in cell culture, supernatant, and in different biological fluids, including blood, urine, breast milk, amniotic fluid, malignant excited, etc. Exosomes are widely distributed, but how to get high quality exosome has become another thorny problem. Exosome extraction methods include ultracentrifugation, densite gradient centrifugation, immunocapture, etc. And each method has its pros and cons. Pros: ultracentrifugation has been most widely used for exosome extractions by virtue of its simplicity and rapidity. Densite gradient centrifugation has the advantage of obtaining exosomes with higher purity using magnetic fields which conjugate with specific antibodies to capture exosomes is another better strategy. Cons: purity of exosomes obtained by this method is not ideal enough. Its disadvantage is that it takes more time and energy, and few exosomes are isolated, but this method is expensive and produces few exosomes.
So what's in the message? How can we decode, how we can crack the communication code of cancer cells?
The long-term view is to be able to actually to cure cancer.
Exosomes, since they're secreted by tumors, are a critical part of the microenvironment.
We think that cancer cells, they make vesicles and they customize them for their own purposes. Tumor is not anymore what we thought it was. They are very good in making their own language and then actually giving out signals that cannot be intercepted by our immune system. And that's what we're trying to do, decode this language that the cancer cells are using.
In glioblastoma patients, you can actually get genetic information about the tumor by taking a serum sample instead of a brain cancer biopsy.
Exosomes can actually give you some information about the content of the cell, and that can tell you something about whether the cell is normal or headed towards being malignant or is fully malignant. Those types of biochemical markers could be incredibly valuable in detecting cancer at a stage where it's early and can be treated effectively.
Just taking some blood is much easier than taking a biopsy.
We have developed a platform in 3 biofluids. So we're working in urine samples as well as blood and cerebrospinal fluid. We're focused on developing diagnostic tools for prostate cancer, brain cancer, neurodegenerative disease, and other solid tumors.
A lot of patients that are diagnosed with a certain disease, they are treated all alike, but not all patients are similar. Maybe the exosomes, they can say something how the treatment worked. Was it good? Did it work efficiently or not? You can use exosomes potentially as therapeutics to directly target cancer cells and then kill the cancer cells without killing the healthy cells.
Currently, there is a program that's being run in France to use exosomes from patients with cancer so to purify the exosomes from their immune cells and to re-inject them into these patients with the hope to boost their immune responses against their own cancer.
So you could load these little vesicles with very specific targeted molecules that only are damaging to the cancer and not to normal cells.
Exosomes used this way would be a sort of personalized therapy.
We have hurdles to get there, but it's an exciting opportunity to actually reach much further in those fields.
How is it applied in Ecuador? Exosome therapy. Recent studies have shed valuable light on the applicable functionality of exosomes. By combining old cells from an aging organism with immature cells from a younger organism, it is observed that the exosomes available in the younger stem cells will take on the role of revitalizing the aging cells. This Highland technique is currently being applied in regenerative medicine. Degenerative diseases are caused by a continuous deterioration of cells, which negatively influence the organs. Stem cells drive cell revitalizations and external influence can obstruct their activities. Stem cells can lose the ability to produce all the necessary data. Help stem cells with external exosomes has a significant positive effect by producing new data to support the healing process. Conclusions. Our findings elucidate not only the presence of exosomes in the epithelium and mesenchymal, but also their roles in mediating epithelial and mesenchymal cells functions. Exosomal tracking between epithelium and mesenchymal through the basement membrane appears purposeful and preferential by reciprocal cell types, leading to specific induced cell functions, including the presently stated cell differentiation and matrix synthesis. Reciprocal exosomal functions and lieu of epithelium or mesenchymal cells suggests that exosomes might augment or substitute classic morphogenic interaction by cell-cell contact and protein receptor interactions.
Direct Biologics was created to expand the science of cutting-edge biologic technologies through research and commercialization of the naturally occurring proteins from allograft tissue, compliant with FDA standards for 361 HCTP product. Cells by nature work to heal, repair, and reconstruct the body. Science shows us a revolutionary way to provide true stem cell therapy, which is to provide therapy to the patient's own cells by modulating the cellular activity. This happens by introducing the patient's cells to a healthy and younger combination of naturally occurring growth factor proteins and exosomes. A growth factor is naturally occurring protein capable of stimulating cellular growth, proliferation, healing, and cellular differentiation. Usually it is a protein or a steroid hormone. Growth factors are important for regulating a variety of cellular processes. Let's find out how exosomes are made. Inside the cell, a cell membrane forms inward, forming a special type of lipid vesicle called an endosome. Then the proteins, microRNA and messenger RNA, invaginate the endosome's membrane, forming the exosomes. Endosomes then fuse with the surface of the cell, releasing the exosomes outside of the cell. Due to the observation of this process, historically exosomes were believed to be a waste byproduct of cells. But when researched further, and it was discovered that, in fact, these are the mediating mechanism involved in direct cell-to-cell communication. These produce an anti-inflammatory response due to the communication from the many proteins that down-regulate inflammatory proteins. Other proteins will then recruit stem cells to the area that will help unregulate these cells to proliferate and promote organized healing through the regulated process. Then once the task is complete, there are additional proteins that tell the cells when the healing has been completed. These exosomes, with the presence of over 1,000+ growth factors, are being regarded as one of the purest forms of stem cell therapy. In their natural state, they offer proteins and no DNA or potentially harmful byproducts that can be found in other therapies.